Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 2 for January 2022. This is the Part 2 video. I'll put the link to the Part 1 video below in the description box. Let's begin with question 5. Question 5. Listeria monocytogenes is a bacterium that contaminates food and causes the disease listeriosis. The photograph shows a colony of bacteria as seen using an electron microscope. So this is the colony we have here. They say the bacterium labeled X on the photograph is 0.5 micrometer in length. Calculate the magnification of this image. So we know magnification should be image size divided by the actual size. All we needed to do is to measure that length X. So when I used a ruler, the length X was approximately 0.8 centimeters, although to some it could be 0.75 centimeters, so about 0.75 to 0.8 centimeters. Since this is my image size, my actual size is 0.5 micrometers, so I have to convert this into micrometer. And to do that, you multiply by 10 power 4 to convert centimeters into micrometer, and my final magnification was 16,000. So the answer should be times 16000. The acceptable answer should be between times 14000 to times 18000. Here they say state why a light microscope cannot be used to view this bacteria. If we go back and look at the size, we will need a lot of magnification to be able to visualize this because it's 0.5 micrometer. That size is quite small to be visualized using a light microscope. So I said it has actual size of 0.5 micrometer, which is too small to be viewed with a light microscope. And also the magnification used in this case is 16,000, so it's quite big. That cannot be obtained or achieved by a light microscope. Moving on. Here they say the photograph shows a bottle of oil extracted from the plant oregano. This plant is native to Mediterranean and Western Asian countries. So here we have the oregano plant extract. Down here they say oregano oil contains antimicrobial substances. The oil can be used in the production of plastic used to package food. Explain why this packaging may be chosen over traditional oil-based plastic packaging. This packaging is going to be biodegradable and sustainable because it is made from plant-based material. It also has antimicrobial properties, so bacterial growth on the food can be minimized or it could be prevented. Also, this will lead to fewer people getting infection from food poisoning or from infections that are caused by bacteria. Moving on. The photograph shows green algae called sea lettuce. This is the sea lettuce. Sea lettuce contains chemicals called phacoxanthins that have antimicrobial properties. The antimicrobial properties of these chemicals were investigated. Discs containing phacoxanthin chemical were placed on 2-4 agar plates, each seated with a different type of bacteria. These plates were then incubated for 24 hours at 25 degrees Celsius and the zone of inhibition measured. The diagram shows the zone of inhibition, which is this, around the material containing the antimicrobial substance. So they say the table shows the results of this investigation. We see here they have the type of bacteria and the mean diameter of the zone of inhibition, and then the range for the standard deviation. We have 10.2 plus or minus 0.72, which is the higher number is that and the lower number. For this, we have 11.1, again plus or minus 0.63, which is 11.73 until 10.47. For this one here, we have 6.0, which is plus or minus 0.41. It could be 5.59 to 6.41. This is 7.5. We see plus or minus 0.55. The highest is 7.75 and the lowest is 6.95. So we can see this is going to be the least affected because it has the smallest mean diameter of the zone of inhibition, while this one is going to be the most affected because it had the greatest mean diameter of the zone of inhibition. Here they say which type of bacteria was least affected by phacoxanthin. If we go back here, and again, the diameter was 11.1, so this is going to be the most affected, and that is the least affected, which has 6.0. So here, the answer should be a C. Next they say, the table shows the mean and the range of diameter of the zone of inhibition for each type of bacteria. Calculate the maximum difference in the diameter of the largest and the smallest zones of inhibition. So the largest was 11.1 plus 0.63, which gives us 11.73. And the smallest should be 6.0 minus 0.41, which gives us 5.59. These values can be got from the table. If I take you back slowly here, we have 11.1 plus 0.63 will give us the largest value, and 6.0 minus 0.41 will give us the smallest value. So the maximum difference should be the largest value minus the smallest value, which gives us 6.14 millimeters. Down here they say drugs containing these chemicals must be tested for safety before they can be approved and used to treat humans. Three-phase testing can be used to check for the safety and effectiveness of these chemicals. Describe the role of phase 1 and phase 3. They test for the possible side effects the drugs may cause, and then also they can be used to determine the dosage or the concentration of the drug that should be used. They can also be used to determine if the drug is more effective than the best existing treatments for that specific infection. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Skin color is an example of continuous variation in some species. In one species, skin color is polygenic and controlled by 3 genes, A, B, and C. 2 individuals heterozygous for each of these 3 genes were crossed, and the diagram shows the possible allele combinations present in the gametes and the offsprings produced in this cross. The shading indicates the darkness of the skin color of the offsprings. So here we can see the darker is the dominant allele present and the lighter is the recessive allele present. Here we can see the 2 crosses, and for the sperm cells, that is what they have, and the egg cells have that. And the number of dominant alleles from 0 until the maximum, which is 6. So you can see, of course, there are more offsprings with darker skin tone than those with lighter skin tones. So moving on. Here they say the graph shows the probability of the skin colors of the offsprings from this cross. And again, we see on the vertical axis probability, horizontal axis number of dominant alleles. So here they say explain why there is a large variation in skin color of the offsprings produced from this cross, and use your answer from the question and your own knowledge to help support your answer. From the graph, the highest probability is for 3 dominant alleles to be present. So here I say it from the graph, the phenotype, which is skin color, shows continuous variation. We can see that this is continuous variation. There is a higher probability of offsprings inheriting 3 dominant alleles and a lower probability of inheriting either 0 or 6 dominant alleles. We can see fewer 0 and 6 dominant alleles. Most of them are going to inherit 3, and the next will be 2 and 4, and so on. So I went on to say, from the table is what we saw here. So the next part is going to be talking about the skin tones and so on. So from the table, there are more dominant alleles found in the offsprings. Remember, the darker colors were the dominant ones, so we saw from the table, there are more offsprings with darker skin color. So more offsprings have a darker skin color. However, because skin color is a polygenic trait, the offsprings have varying tones of skin color. You saw that as well here. They are varying tones. Not everybody has the same skin color. They are varying tones, and therefore, that means there is a combination of alleles that are playing part in the skin color, which is the phenotypic trait. Then I say the great variations in skin color could be due to crossing over in prophase 1 and random assortment of metaphase 1. And this lead to formation of gametes with various combinations of alleles of the skin color. Here you have to remember that prophase 1, there is crossing over, and that causes sharing of specific genes between non-sister chromatids. Also, the random assortment occurs and various combinations could result out of that. Also, because fertilization is random, various combinations of gametes can fuse, leading to zygotes with different genetic combinations for the genes involved in determining skin color. And finally, skin color can be controlled by environmental conditions like increased exposure to the sun, as well as mutations that could occur, and these could all increase the variation that we have observed among the offsprings. Down here they say, suggest 2 reasons why individuals that have inherited the genotype AA, BB, CC may have a darker skin color than other individuals with the same genotype. If individuals have the same genotype, I'll say like identical twins, and they have different phenotypic characteristics, it is because the environment has played a specific role, or it could be that a mutation has occurred. So I said mutation in genes that determine skin color can result in genetically identical individuals having different colors. Also, exposure to the sun leads to a darker skin tone. Then other molecules from the environment, or could be from food, could affect cells involved in skin pigmentation, resulting in different colors. So this brings us to the end of question 6. Let's continue to question 7. Question 7. The photograph shows a Titan arum plant, which we see here. They say this plant is native to Sumatra, Indonesia, and is critically endangered. There are fewer than 1,000 individuals in the wild and 500 in botanical gardens around the world. Sumatra is described as having a high species richness. State what is meant by the term species richness. This is the different number of species found in a specific habitat. Moving on. So here they say seed banks often conserve endangered plants by storing samples of their seeds. The seeds are dried and then frozen. Explain the advantage of drying seeds before storage. Now, we know the process of drying removes water, and because water is a requirement for germination to occur, when water is removed, the enzymes that participate in the production of glucose or sugars that are used in germination will not function properly, and that means germination can be delayed. So I say drying removes water, and this slows down digestion of the food stores inside the seed by enzymes. This prevents the seed from germinating during storage, and as a result, the storage life of the seed can be extended. Moving on. Here they say, scientists have stated that more than a third of critically endangered plant species cannot be saved from extinction by storing seeds in seed banks. The Titan arum is one of these plants. This plant flowers once every 10 years, and the flowers last for 2 to 3 days. The plant can reproduce asexually or sexually. In order to reproduce sexually, it relies on insects to transfer pollen to other Titan arum plants. Scientists are developing ways to conserve this plant without losing genetic diversity. Some of the suggestions include grow more plants produced by asexual reproduction in botanical gardens, collect pollen when an individual plant flowers and store it in a seed bank, create a stat book for the species, and artificially pollinate the plants in the wild and in botanical gardens. The first part says the heterozygosity index can be calculated using an equation. Write this equation. The heterozygosity index is equal to the number of heterozygotes divided by the total number of individuals in that species. Next, they say the heterozygosity index of 500 Titan arum plants in botanical gardens was found to be 0.166. Calculate the number of heterozygotes in this population. The number of heterozygotes is going to be that, so which is going to be that times that. In this case, it's going to be that times that, and the answer comes out to be 83. Moving on. Here they say, discuss the suggestions proposed by these scientists for conserving the Titan arum. If I take you back here, you can see these were the suggestions, so I'm going to discuss one at a time. The first one was growing more plants produced by asexual reproduction. We know this will lead to an increase in the number of plants, but they will be genetically identical because remember, this is reproduced asexually, so they will be genetically identical, and this will lead to offsprings with less genetic diversity, and so less chance of survival if conditions change. Changing conditions could be like if there is an infection, or the plants that are genetically identical have a higher chance of dying, so extinction could occur. Next, collecting pollen when individual plants flower and storing it in a seed bank will lead to availability of more pollen when needed, and there can be a chance to use pollen from different areas during artificial insemination. There could be low genetic variation if pollen from the same plant is used to inseminate more plants. Also, the cost of storage of the pollen in seed banks may be high, yet viability during storage may not be guaranteed. This is viability of the pollen during storage. If it cannot be guaranteed, then there is a chance that this could not work. Using stat books for species allows to keep records of the genetic information of individual plants so that decisions made during selective breeding can lead to increase in genetic diversity. Stat books are used to store information of which types of organisms have been crossed in order to avoid crossing the same organisms in the future if your aim is to increase genetic diversity. Lastly, I say that artificially pollinate plants in the wild and in botanical gardens could lead to more successful pollination, resulting in population increase, since less time is available for flowering in the wild. Pollen from individuals with different alleles can be used to increase genetic diversity. So this brings us to the end of question 7. Let's continue to question 8. Question 8. Gametes are specialized for their function. Egyptian female desert ants mate with more than one male. A male desert ant has an adaptation to improve the chances of his sperm fertilizing a female's egg cell. The sperms are released in bundles, with their heads held together by a sticky protein. The diagram shows a bundle of desert ant sperm. So the first part suggests how the sticky protein would have been secreted. This is talking about secretion and not making the protein or protein synthesis. It's just talking about secretion from the cells in which the protein is made. So, for the secretion to occur, it would have been packaged by the Golgi apparatus into secretory vesicles, and these vesicles would have fused with the cell surface membrane to secrete the sticky protein outside of the cell. Here they say the graph shows the speed of individual sperm compared with bundles of sperm in liquids of different resistance. So here we see speed of the sperm, and down here as resistance is varied. Our observation is we can see the individual sperms are always at a lower speed in comparison to the sperm bundles, and we see that generally as resistance of the liquid increases, there is a decrease in the speed of the sperm, but the individual sperms have greater decrease from 0.5 in comparison to the sperm bundles. So here they said comment on the results of this investigation. So here I said sperm bundles have greater speed than individual sperm at all resistance of the liquid. We can see that at all resistance they have a greater speed. This could be because greater force is generated with sperm bundles than with individual sperms, because the bundle has a lot of tails close to each other, so they are propelling, and as the propulsion goes on, greater speeds can be created or greater force can be created, meaning they will have greater force as they move through. Then, increasing resistance of the liquid above 0.5 AU decreases the speed of the sperm, but there is greater decrease with individual sperm than sperm bundles. And lastly, from the data, presence of error bars shows that the error bars are these ones here. It shows that the repeats were carried out, the mean was calculated, and so the results could be believable. However, there is overlap at 0.5 AU for both sperm types, so there is no significant difference in the results. I'm talking about the results here. The highest for this and the lowest for that do overlap, so this result here is not dependable. Moving on. Here they say the photograph shows an Amur leopard found in southeastern Russia and northern China. In vitro fertilization, IVF techniques are being developed to help conserve this endangered species of leopard. These techniques are being developed using mice. IVF involves the removal of egg cells from the female mice. The zona pellucida can be damaged during this process. The effect of damaging the zona pellucida was investigated. 300 mouse egg cells were distributed equally in 3 petri dishes containing nutrient solution. Samples of sperm of different concentrations were added to each petri dish. The volume of each sample added was the same. The experiment was repeated for egg cells which had their zona pellucida damaged, and the graph shows the percentage of egg cells fertilized by more than one sperm, which is the percentage of polyspermy. So here we can see percentage of polyspermy with those that had intact zona pellucida and those that had damaged zona pellucida. This was not there. I drew that. So we can see, except for this, where the concentration was lower, the higher the concentration, the greater the chances of polyspermy. But those that had a damaged zona pellucida have greater chances of polyspermy. So here they say when the highest sperm concentration was added to the egg cells with damaged zona pellucida, the percentage polyspermy was 52%. Plot this result on the graph. Now this is 50, and you can see that is 60, meaning that small box represents 10%. If it represents 10%, then each small box represents 2%. So 52 should be around here, one box higher than 50. And if we can look at all the other bar graphs, you can see if 3 squares horizontally, I also drew 3 squares horizontally, and I left 2 spaces here, like they left there, as well as there. So you have to be consistent with what you have been given. So that was my answer. Next they say explain the result of this investigation. So I have to explain, and remember this is a formal question, so I have to write appropriately. I say there is a higher percentage of polyspermy of eggs with a damaged zona pellucida. Due to the damage, cortical reactions could not occur in the damaged parts of the zona pellucida, so those areas could not be hardened to prevent polyspermy. There is a higher percentage of polyspermy as the concentration of sperm increases. I hope you can see that. Higher concentration from 50, which is that, 100, which is that, and 200, which is that, for all. As the concentration increases, there is a high chance of polyspermy. This is because increased sperm concentration can lead to a higher chance of more sperm reaching the egg cell and entering at the same time before cortical reactions can occur. So this brings us to the end of question 8. Let's continue to question 9. Question 9. The Hood Island giant tortoise is only found on one of the Galapagos Islands. The photograph shows 2 Hood Island giant tortoises fighting over territory. These are fighting over territory. You can see they have long necks. They say which of the following is a term that would be used to describe the fact that this species of giant tortoise is found only on the Galapagos Islands. The answer should be B. It's endemic to those islands. Part B. The Hood Island giant tortoise has adaptations to its environment. 2 features are: they have long necks, shell that arches above the neck. When there is a dispute over territory, the tortoises use their features to make themselves look as large as possible. Complete the table to show the type of adaptation shown by this tortoise. So the first feature is long neck. This is anatomical. Shell that arches above neck. This is also anatomical. In making themselves look as large as possible, this is behavior. Next, they say suggest one selection pressure that results in the development of one of these features. These features could have developed because of competition for food or competition for mates. Moving on. Part C says, in 1963, there were only 3 males and 12 females of C. hudensis left in the world. These 15 tortoises were used in a breeding program. The diagram shows the number of offsprings from each female tortoise and the fathers of these offsprings. Observing the graph, vertically we have the number of offsprings and horizontally we have the females. Here we have the fathers A, B, and C. They want us to calculate the percentage of female 10's offsprings that were fathered by male B. So female 10, these are the offsprings. These were fathered by father B, and these were fathered by father A. No offsprings of female 10 were fathered by father C. You had to get the representation of that. This is going to be 18 because when you look at the scale, a tiny box represents one member or 1. So these are 18 boxes. That means it's going to be 18. So the percentage should be 18 divided by the total. The total is 27 times 100, which gives us 66.7%. Down here they say the white population of C. hudensis is now 1,800 individuals. Calculate the percentage increase in the white population of C. hudensis tortoises and give your answer to 1 decimal place. The increase should be, remember right now it's 1,800, but initially there were 15. So 1,800 minus 15 gives us that. And then 1785 divided by 15 times 100 gives us that. So that is going to be the answer. Moving on. Here they say a zoo tested its captive bred giant tortoise and determined that it was a C. hudensis tortoise. Explain how the zoo determined its giant tortoise was a C. hudensis tortoise. To do this, they should have the DNA of the C. hudensis, and then they should also get the DNA of this tortoise and compare. So I said they analyzed the DNA of the zoo tortoise, and then they compared this with the DNA from the actual C. hudensis, and then they bred the zoo tortoise with a C. hudensis to see if the offsprings produced are fertile. Lastly, here they say scientists thought that a recessive allele was changing in frequency in the tortoise population. Explain how the change in frequency of this allele could be determined. They have to carry out DNA analysis to identify the different alleles present and use the Hardy-Weinberg equation, which is this one here, and then compare the recessive allele frequency of the current generation with that of the previous generation. If they are different, they can make a judgment that is changing, or if they are the same, then it's going to be constant. So this brings us to the end of question 9, as well as the end to this paper. This was the second part of the video of the paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.